Professor Dr. Mark Bühler, who works for the Novartis Research Institute in Basel in Switzerland. And he was also attending the scientific conference. He's made a major contribution into our understanding what the ADNP protein does in the body. And he will now tell us a little bit his thoughts about how, how we as researchers or how industry thinks about what we can do with drugs for this type of diseases. So please go ahead.
Thank you, Frank. So Frank gave me the difficult task to talk about approaches to target the genetic cause. So it's difficult because it's difficult to do that. So I'm not gonna, report about a successful research project where we could say, yes, we can do it. So in 5 years from now, we would have a therapy. So you should not have too high expectations. What I try to do is to explain to you what the mutation in the ADNP gene, what it really does, and how we could think of potential therapeutic approaches to correct that mistake nature made by chance. I'm aware that most of you are non-scientists, so I try to use lay terms and not use too much jargon. Maybe it's too trivial, so then I apologize. Maybe it's still too complicated, and I apologize as well. So if that's the case, please don't hesitate to interrupt and ask if you don't understand. Okay. So before I start, I just want to quickly tell you where I come from. So I'm a professor at the University of Basel in Switzerland, and I'm leading a basic research group at the FMI, also in Basel. So FMI is somewhat special in the academic landscape. So we are a research institute that is basically, you can place at the intersection of industry and academia. So FMI was founded about a bit more than 50 years ago by a company called Ciba-Geigy. This company later merged with Sandoz to become Novartis. So Novartis you probably have heard about. And for all those years, the founders of Novartis has been very generous in supporting us financially. So this is a kind of a contribution to society. We are free to operate, so we are academic scientists. Nobody tells us what we have to work on, but yet we have this connection to the pharmaceutical industry. So our daily life is like all the other professors in the room. We are training students, we have PhD students, and we can take risks. So we can work on risky projects. A company like Novartis would never engage. And unfortunately, that's the case still for those multi-founder syndrome. We don't know enough. It would economically not make sense to put $1 billion on such a project. But we are free to do this, and we do this. So I'm doing this because I think the ADNP gene is a very interesting one, and I try to explain you a little bit why I think that is the case. And I want to start with showing you this slide, which I also showed yesterday already. So I grew up in a valley in Switzerland. So my grandparents, they had a farm, they were breeding cattle. My father was breeding rabbits. When I was a little boy, I was really happy to have all these pets around me. But the older I got, the more I started to realize that how animals reproduce is really a miracle. You imagine it's two cells that come together, one from mom, one from dad, they fuse, and then an entire cascade of cell divisions is triggered. An embryo is growing until you have a fully grown animal. So in our case, it would be a baby that is born. And a lot of things are happening during this process. And I'm saying it's a miracle because so many things could go wrong during this process. And yet, in most cases, things are very accurate. That's why we are all here sitting in this room. But sometimes, not everything is okay. And I don't have to tell you that. So we have about 20,000 genes, and the ADNP gene is just one of them. And it takes just one letter that needs to be wrong in the ADNP message, as we heard this morning, and then you will see something like health modifier. So that's why I'm so fascinated by biology. And this is not only humans; it's basically everywhere, even in plants. You see all these very complicated processes. And we try to study this. We try to better understand this. And it just turns out that the ADNP gene and the protein is very poorly understood. And we would like to much better understand how this is involved in processes like this. So what you see here is different organs, different tissues, and it's important to mention that all the cells, whether it's in the brain, whether it's in the heart, in the muscle, they have always the same set of 20,000 genes. We carry that around with us all the time. But you don't need the same genes to be active in the brain as you need them to be active in a muscle. And this is a very important process. And there is proteins that do direct cells into the right direction. So you can imagine that you have progenitor cells. We call them also stem cells. So also adults have that. And this is illustrated here by this cell here. And then they get cues, they get stimulated to become something different. So stem cells then can go down, let's say, this valley here, and they would become a neuron. Or if they would go down this valley here, they would become a muscle cell or a blood cell. All the different cells that you find in the body. And from the research that has been done so far, we learned that ADNP is probably playing a very important role in making such decisions. So normally, when a stem cell tries to go into a certain valley, this is done very accurately. And ADNP seems to be important for that. If you have a mutation in ADNP, somehow these cells are confused. They are kind of a bit desperate. They want to run, and then sometimes they go down the right route, but often they go down the wrong path. And this is more or less a bit a stochastic process. So, once it happens like this, once it happens like the other way around. And maybe this is one part of the explanation why you are observing so different problems, issues in your children. Because not, as we heard, not everyone is doing this. But much more needs to be done on that to really fully understand what's going on. So now I try to explain you what this mutation we always talk about, what is this really doing? And for that, I would have to give you a little bit of a molecular biology lecture. And maybe you know all this, but I do it anyways. So we heard from Claudio already that our genes, they are made of DNA. So we have, as I said, in every cell, we have the entire set of the 20,000 genes. And basically, the molecule called DNA is the code for everything we need to do. And it's quite amazing. So this molecule is about 2 meters long, which fits into a tiny little nucleus of our cell. So I can compare this usually when I try to explain this to my parents, is that you can think of a gene like a recipe. So ADNP would be a recipe for a Black Forest cake. You know Black Forest cakes? You like it? Me too. But we have not only just one recipe, we have about, as I said, 20,000. So our genome, these 2 meters of DNA, is basically your grandma's cooking book. So you can think of the cake as being the product of these recipes. So if you go to the kitchen, this recipe tells you exactly what you have to do that you end up with a nice Black Forest cake. And then you may eat this, and if you follow the recipe correctly, you will enjoy it, hopefully. So like this little girl here, you see the smiley face, so she really seems to enjoy this. And that's what we scientists refer to as a phenotype. So this cake is doing something to this person. Now, there is something else written here. This is ADNP mRNA. So this is a very precious book because you got this from your grandma. So you really don't want to make it dirty. So you don't bring this to your kitchen because you could spill something over it. So it's better to first make a copy. If you want to make a cake, then you copy the ADNP recipe, you bring that to your kitchen, and it doesn't matter whether you spoil something or spill something on it or not. And this is what our cells also do. So the gene itself is not being translated into the protein. So it always first needs to be copied into one of these copies, which we call mRNAs. Okay, so it cannot happen that one of these letters here in the genetic code are different. And of course, that then can have an influence on how you read this recipe. No, it's like if you make a typo in your recipe, you may then start putting salt instead of sugar into this cake. And you can imagine that the phenotype would be a bit different. So that girl will probably not smile anymore. Maybe that would even cause a gagging reflex. I don't know, something else. And that's how you can think of the mutation that your children have acquired. So the cells, they have a recipe for this protein, but they basically make something that makes the protein not function. And what is this mutation actually doing? So the RNA again itself, this copy of the recipe, is not doing anything. It's still just an instruction to make a protein. And there, our cells, they have machines. They're drawn here. We call them ribosomes. So these are the machines that can read the code on the mRNA. So this is basically the chef who is reading the recipe and basically would then build or cook the dish or the cake in my example. Now, the ribosome needs to know where it has to start reading. And there is very specific three-letter codes, which tells the ribosome that this is the start of the recipe. So you should start here, and that's indicated here. And then it would hop always in steps of 3 letters, and always the 3 letters encode for another, building block of a protein. So it's moving on along this mRNA, and is adding one brick after the other, and that's how a protein eventually is made. So the ADNP protein is made like this until there is another 3-letter code which tells the ribosome, now you're done, so you can stop here. So the ribosome will stop here, and then you get a protein that has a certain length. And for ADNP, this is a bit more than 1,000 of these building blocks. And depending on how these building blocks are arranged, proteins then have a different activity. So now if you have a mutation, and in many cases, and we heard this morning, there is a hotspot of mutation. So in that case, for this hotspot, the famous 719, tyrosine 719, there is one letter change. There's just one of these letters is changed, so that in the middle of this sequence, there is now one of these stops created. So the ribosome now is told that it should stop making the protein here, and it will no longer go to the very end. So basically, the chef would stop in the middle of baking this Black Forest cake. So maybe you forget to add the sugar, or you don't bake the dough or whatever. So obviously that would not be good. So why is that bad? So why is this not good for the cells? Well, we still try to understand why exactly this is bad. But what we know for sure is that we have now a protein that is missing some of its part at the very end. So now I have to tell you that ADNP has friends. So ADNP is talking to other proteins. And it turns out that it's talking to several proteins, but probably many of these interactions are just small talk. So they talk to each other, but they're not very, very, very good friends. But ADNP has got two very good friends. I think they even have a very intimate relationships. They always hang out together. So it's CHD4 and HB1, and you don't really need to know what they do. But it's important to know that these three proteins, they form a functional link. And we now try to understand what is this. So this we also refer to as the CHAP complex. You may hear about this again if you read the internet about ADNP, but it doesn't really matter for today what this really is. I just want to mention this because we have a situation. So in all the parents, you basically have this situation. So we always got one of these genes from our mom and one from our dad, right? So we always have two. So basically, you would make two copies of these ADNPs. So we would also have two of these CHAP complexes. Of course, we have many more, but in principle, it's two, one from our mom and one from our dad. Now, in the patients, the situation is such. So we have one of functional ADNPs. There is no mutation. So we are making functional CHAP complex. So it can function normally. But then from the other, what we call allele, from the other gene, we have this mutant form. And basically, this is creating something that looks like this. So it still interacts with this yellow protein, but is no longer able to interact with the protein that we call HP1. So what could that now mean for a cell? So here we're still not so sure what we would think about this, but scientists distinguish between loss and gain of functions. And you may have heard about this. I'm not so sure. I try to explain this. So in the case of a loss of function, it's basically it just means that the ADNP protein that is mutated can't do anything that it normally would do. So it's just useless. Which means that you just have half of your functional ADNP CHAP complexes, and the rest is just there and not doing anything. So this is what scientists refer to as loss of function mutations. So if you think about the cake analogy, this would be something like putting less sugar into the cake, just only half. So this would still be somewhat edible, would still be tasty, but the phenotype probably would already be a bit different. You could also think now of having a gain of function. And this is a situation where the actual mutant version is doing something that it is normally not doing. And you can again think of this maybe adding salt and sugar into the cake. Huh? So the salt, the sugar would come from the healthy one, and the mutant one would be salt in the cake. And again, this will be something that doesn't taste very good. So now, if you have just half of it, you could think of therapies that just promote the production of this protein. And in the cascade I showed you, you could interfere at any step. You could make more mRNAs. You could try to make the mRNA better translated into the protein, all things like this. In this situation, you could think of an approach that would try to destroy this mutant form because it's bad. You don't want to have it. So you can again could think of ways to remove that from all. These would be valuable approaches, and depending on whether it's gain or loss of function, you would do the one or the other. But in the best case scenario, you would try to restore this situation so that you could revert this mutation, so that you could directly attack the mutation, and then it wouldn't matter what it is. So then these scientists would not need to debate whether it's gain or loss of function. If you would just be able to correct the mutation, then we would be fine. And I would like to illustrate you how one could think about doing this. And this is basically precision medicine. It's something very fancy. It's being done in very few cases, but it's not a common drug that you would buy these days. So the first thing you can think about would be a gene therapy. So what does that mean? So this would mean that we would directly act on my grandmother's recipe book. So we know there was a mistake in this book, and we now go and we edit that mistake. So that whenever we make a copy again from that book, we will always have the correct recipe. So for a long time, we could only do this in some weird model organisms that scientists use, like yeast you use to make beer in Belgium, or bake bread. Everyone is using yeast. So we could do this. We could modify the genetic code of this organism. In animals, this was really hard to do, if not impossible. But you may have heard about the CRISPR technology. So just a few years ago, it was again academic, crazy scientists who worked in bacteria, and they found that they have molecular scissors that can cut DNA. And they could show that we can use these bacterial proteins in our cells to also cut DNA. And by doing this, we can repair mistakes. So this would be one way to think about correcting the mutation in the ADNP gene. But this is a very immature technology and is associated with many complications. But if you think about doing this, for example, in the hemopoietic system, so the tissue that makes blood cells, that's something you could start thinking about, because you can take bone marrow, you could manipulate cells, and you could bring back the stem cells, and then you would have a properly, normally functioning immune system again. So that would be one approach one could start thinking about. So another thing people thought about already is trying to fool the ribosome. Tell the chef that he should not listen to that stop codon. He should just ignore it and keep making the protein. And here, it has more profound patients would be in good company, because it's a rare disease, yes, but the actual cause, these stop codons that are created by these mutations, are very common. So it's estimated that there is about another 2,400 diseases that are also caused by such stops. Of course, not in the ADNP gene, but in another gene. So famous examples are Duchenne, for example, or cystic fibrosis. Also there is a stop codon created that should not be there. Of course, people notice this early on, and what they try to use is drugs, low molecular weight compounds, that would basically tell the ribosome that don't care about that stop, just keep going. And one class of molecules that is doing this are aminoglycosides. So these are actually antibiotics. So they have been developed to kill bacteria, because also bacteria, they have to make proteins. They also have ribosomes. And these ribosomes, they stop making proteins. So that's why they can kill the bacteria. But in our cells, our ribosomes, they still cook or bake, but they would not listen to these stops anymore. So we have tried this in our lab. So we have been working in a cell culture model. So this is in cells in addition, where we have cells that express this ADNP with the 719 mutation, so with this stop codon. And you can see, as I showed you before in the schematic, it interacts with this protein here, CHD4, but we don't see these HP1 proteins. Just cannot detect it. And it's because the ribosome stops before it translates a part of the protein that likes to grab the HP1s. Now, when we add these antibiotics, so we tested two different kinds, you can now see that the HP1 proteins are coming back. So this shows that this is a valuable approach. It's proof of principle that if you give cells the instruction that they should not listen to these stops, ADNP is now made full length, even though they have a mutation. Now there is a problem. This is not only happening on the ADNP mRNA. So any other RNA that also has a stop codon, the ribosomes will also not listen to the stop. And you can imagine what could happen. And I think this is one of the biggest problems, not specific. And that's why the aminoglycosides have been dismissed as a therapy. So for long-term treatment, this is definitely no option. You may think of using this as an acute response, but you definitely don't want to take this forever. Okay, so how could we then do this more specific? And again, I'm really fantasizing here, fantasizing the term that it's difficult to develop this, but technically it's possible, and that's what I'm going to show. So instead of fooling the ribosome, we could also take the photocopy and correct it. Those of you who are old enough who may still remember typewriters. Now, if you made a mistake, you had this ink, Tipex. You could Tipex it, and then you would type the right letter. So that's what we could do. Actually, our cells do this all the time. So they do edit RNAs. So these copies, when they are made, and there in the cell we have an RNA editor, it is making changes to the code on the RNA. The DNA, the gene is not touched. So there were people thinking about this, so trying to use our endogenous editors and tell them where they have to make edits. Because the editor on its own doesn't know where to edit, it needs to be told. And the very cool thing, at least I think it's cool, is that you can program the editors yet with another RNA. So in this case, the RNA is not a recipe for a protein, but it basically contains the address for a certain gene. So you can make an RNA molecule which knows how the ADNP mRNA looks like. And this will help the editor called ADAR find this position on the ADNP mRNA, and it would correct that mutation. Okay, so we have done this in the lab, just to show that in principle this would be possible. This is the RNA molecule that we use to program the editor. And this looks horribly complicated. And it is horribly complicated. But it took us a while to design this to really make it work. But here I show you that it indeed works. So this is how a sequencing profile would look like. So we are sequencing the ADNP mRNA. So this is mRNA from a mutated ADNP gene. So you can see every color here is representing one of these four layers. So green is a T, black is a G, and then in red you have an A. And this over here is the stop codon. So these three letters, T, A, G, they tell the ribosome you have to stop here. So that's our problem we are dealing with. Now we told the editor to change this A here into a G. And you see that it works. So you see here peaks for black and red. And they're about the same height, which means we get an efficiency of about 50%. So 50% of our mRNA molecules, they have still an A, but the other 50% now has a G. And this codon now here, this triplet, TGG, is coding for amino acid, one of these building blocks for a protein. So the ribosome no longer thinks it should stop here, and then it keeps going. So this is in cells. We haven't done this in animals. We don't know whether it's applicable in animals, but I think at least it shows that this would be another way how to think about we could design a precision medicine. And this would be specific for the ADNP mRNA. And we would not affect all the other genes that are important for it. We don't want to affect all the other residues. If I would have talked about what this would be, this would be an RNA therapeutic. So we would deliver an RNA, so that the drug would be the RNA that tells the editor where to edit. 5 years ago, people would have laughed at Most people thought this is stupid, you cannot do this. But there were some biotech companies like biotech or Moderna, and that might ring a bell. So I assume many of you got vaccinated about 3 to 4 years ago, or maybe 3 years ago. And if you got the biotech Pfizer or the Moderna vaccine, you basically got injected recipes. So you got injected mRNAs into your arm. And these mRNAs did contain the recipe for the spike protein of the virus. And that then mounted an immune response. That's why we then became immune to the SARS-CoV. So it's possible. So you can deliver RNAs into cells, into the human body. And I think these days now people don't laugh at you anymore. So actually companies invest a lot of money in developing this for cancer drugs and so on. But of course, there are still problems. So this was delivered to our muscle. The problem is, how would you deliver it to the brain? How do you deliver it to the heart? You know, what you basically would like to. So this might be probably the biggest challenge. If it works, then you have to bring your molecule to the right. Okay, I hope I could a little bit illustrate what the mutation really does. I hope I could give you some hope, at least, that your children are not alone. So there is many other diseases that are also posed by the same type of mutation. And if many scientists work on each and every rare disease, now if they find out how one could, for example, promote read-through of stop codons in a specific way, everyone would benefit. So if you think about really approaching the genetic cause of the disease, we have to think about things like the therapies proposed. So gene therapy would be one approach. So if you are able to correct already my grandmother's cooking book, if that's possible, we can talk about the cure of the disease, right? So the recipe is then fine. So if you do this once and ever, you will have solved the problem. The second part I was talking about with this RNA therapeutics, it would be something that you have to constantly administer. So this is a treatment. This is not a cure. So this would need continuous application. Good. I think I would like to stop here. Working on these projects is interesting from a basic science perspective. So I didn't talk about anything about this today. But for us scientists, it's very exciting to work on the chat complex because it's really revealing a lot of novel biology, even things that we didn't know before, so we would not have studied it. But of course, also being here, meeting you, talking to you, as Claudio said, is giving us an extra boost, an extra motivation to invest time and energy into further studies. These are people that have been working now and in the past in my lab. We have lots of collaborators. I'm not going through all these names. But I would like to acknowledge our funding sources because this is one of the biggest problems a basic researcher is facing. So I would like to acknowledge the NCCR RNA disease. So this is a big consortium in Switzerland where RNA scientists come together it's about 14, 15 labs. They all work with RNAs and they did sponsor our efforts towards the RNA editing. And then I should not forget to mention FMI. So as I said in the beginning, we have received generous support from the Novartis Research Foundation. I think it's not a given that a pharmaceutical company is just sponsoring something that most of the time doesn't we just generate knowledge and nothing that would basically be of economic interest. That's why I also wanted to say. Thank you very much for listening, and I would be very happy to explain you further if you got confused.
So much for really perfect overview of how you think of this. Are there any questions? We have maybe time for a few quick ones. Because
There it is.
the next is already waiting, but he's online, so it's a little bit more difficult to control.
It'll be quick. Thank you. So thank you very much, first of all, for the great presentation. So for the RNA therapeutics, does it also work when you have basically one non-functioning ADNP producing cell, so you have one not producing any protein, or does it only work when you have the mutant ADNP cell, if I can call it like that? Thank you very much.
Yes, so if you have an non-functional copy of the ADNP gene, then I think you can forget about this, what I call gain of function. So you don't have a toxic protein. So in a way, that's already good because all you need to do is to come up to correct the dose. So you just need 50% more. And this you can do by many ways. You can activate the endogenous healthy copy of the gene. But you asked about RNA, very good. Of course, you could also think of delivering mRNAs. You could actually deliver ADNP mRNAs instead of a gene. And you may also then achieve, that you correct the dose, that you add the correct amount of sugar into your cake. Yeah. Exactly. So people like this because RNAs are very unstable. So when you got the vaccine, people they were freaking out. We are getting injected genes, and it's dangerous. This could be dangerous if you inject the DNA. DNA is very stable. It could eventually maybe insert even in your genome. You don't want that. So RNAs don't do this. So they have a short half-life. After a day, they are cleared from our cells, and then they are gone. If it would be doing something bad to you, you could stop the therapy. So that's why some people think it's a really important approach because it's kind of reversible.
One for me, if I may. Are there any plans to introduce those tests to animals, mice, for example?
Yeah, definitely. So we would be very interested in now testing this in a mouse model. So the editing that I showed you. The problem here is really the delivery. So delivering RNAs directly to a mouse is difficult to do. But we can also make genes, transgenes, that code for these RNAs. It's the same principle. So what we will try to do is make vectors, maybe a virus, that we can infect the mouse with, so that the mouse is starting making these therapeutic RNAs itself, and then test whether this would correct the mRNA. And if it does, we can ask, are all these phenotypes, can we see rescues of these phenotypes? Thank you.
Thank you very much. Very, very quick question, because the last one is already, the next speaker is online and needs to.
I'm Swiss, but I don't speak French very well.
But there is two ADNP mutation on the gene, so you can edit both.
In principle, yes. It would just make our task a bit more complicated. We would need to design two molecules that work on both. But anyways, that's something one would need to do as well, right? It's not always 719. We know there is many other positions in the mRNA that can also be mutated. But that's the nice thing about the RNA therapeutic approach, because we can intelligently design the molecule. This is different from what we will hear about later. Normally drugs are, compounds are screened for an effect, and often you don't really know how they act, just they do something. But you often do not start by designing a drug. You try to find one that works, and then you are further refining.
Thank you once again, Small. Wonderful. I'm sure there are many more questions, but we have now an online guest. Dr. Olefson, can you hear me?
I can, yeah.
Yeah, we can hear you.
Can you see my full screen or?
Yeah, we can see your full screen. So thanks very much for joining and apologies for running late a little bit. I'm going to switch the camera in a moment so you can see who you're talking to. Thanks so much for taking the time to update us on your on the ketamine trial, which you have done in the past, and you updated us on actually in the first meeting 2 years ago, and apparently you're doing a continuation. So the parents here are very eager to hear your results. Can I give you the floor?
Yeah, absolutely.
Okay,
I'm speaking to parents. Morning, everybody. Oh, lovely.
there we are.
So that was a very nice introduction. Could have been overly optimistic one because I don't have an update. Sadly, I would talk about our experience today and our plans for the future. Those plans at the moment are awaiting results from several grant applications and various funding efforts. So the idea is that we will move forward, and I'll describe how we intend to move forward, but I don't have any updated results. But I'll give you the ketamine story, and I'm happy to answer questions about it. So you guys, I assume, have been learning a lot over the course of this conference, and probably already at this point know that ADNP syndrome has been linked to glutamate dysregulation. Glutamate, of course, is the critical neurotransmitter in the brain responsible for learning and memory, for synaptic plasticity. Alana and her team has done some nice mouse modeling showing that in mice missing a copy of their ADNP gene, related genes, of their ADNP gene, you see actually decreased expression of a specific glutamate transporter gene in the hippocampus, which is the kind of memory side of the brain. Compared to wild type or typical mice, including mice treated with NAP, which is that ADNP snippet, which I'm sure you've also learned about in this conference. This is a nice kind of preclinical evidence linking ADNP deficiency to glutamatergic dysregulation. And then kind of taking the next step forward, you think about the relationship to ketamine. And the first study here on the top left shows that if you silence the ADNP gene, you actually induce cancer growth in animal models, ADNP being a transcription factor that turns genes on and off. Importantly, then ketamine increased expression of ADNP and then reduced the tumor growth. So kind of a cool disease mechanism there. And then in the second study, it shows that low doses of ketamine can actually prevent the toxic effects of higher doses of ketamine in brain cells. So the idea being that there's obviously concerns about what we've learned about ketamine over the years. It's a approved treatment for pediatric anesthesia, but high doses can be quite toxic. And there does seem to be accumulating evidence now that in this study, the subsequent study at the bottom, that pre-treatment with low doses of ketamine can actually prevent the ketamine toxicity that we see, at least in animal models. And then also the third study showed that ketamine enhanced increased expression of ADNP in brain tissue. So that was another important finding. And all this kind of pointed us in the direction of thinking about ketamine for ADNP syndrome. Of course, we know that ketamine is an NMDA antagonist. NMDA is one of the glutamate receptors. It's blocking this NMDA receptor, but it's doing so at the presynaptic neuron around the idea of inhibitory interneuron. So if you block an inhibitory neuron, you actually increase the release of that, which is what that picture shows. So it also increases a whole variety of that ketamine does of other nerve cell growth factors. So that, nerve cell growth factors promote, you can imagine, nerve cell growth, and they induce synaptic plasticity. So this is an important mechanism of ketamine. And then again, as we know, ketamine at high doses can be quite toxic. Low doses are probably very safe, and low doses can actually, pre-treated with low doses, can actually prevent toxic effects of higher doses. So we set upon this open label study. Open label meaning there was no placebo control. This was a one-time infusion intravenously of 0.5 milligram per kilo. We looked at effects after 1 day, a week, 2 weeks, 4 weeks. Again, there was no placebo condition, so that's an important sort of methodological flaw to keep in mind. We did this just because we wanted to see mostly around safety. So do these kids tolerate ketamine? Really, this is one of the first, I think maybe the first study in ketamine in a neurodevelopmental disorder in children. So it's definitely, we had some concerns around safety. We wanted to make sure that it was safe before we would plan any additional studies. And so we did find that it was quite safe. In fact. So our sample was only 10 kids. There were 3 girls and 7 boys. The kids were, they're 9. I think the age range is 5 to 12, and the average was about 9. So pretty wide age range. Not that it's so critical, but for the kids met criteria for autism, 7 met the criteria for ADHD. So this is kind of what we typically see with the phenotype ADHD syndrome. I think you see on the right side in the table all the different adverse effects. So we don't know really how many of these adverse events were actually related to ketamine, but we wanted to make sure to document really everything we saw. I would say, based on my experience, that probably the first 2 or 3 perhaps were likely related to ketamine, and especially being kind of elated or silly. And, ketamine is a pretty powerful antidepressant, that there's a logic behind that. A few of the kids got a little more aggressive. There was some sedation, appetite changes, anxiety, and then everybody else was sort of minimal effect, right? So, 1 or 2 kids had some irritability, some nausea and vomiting. There are a lot of different side effects, but nothing that was especially concerning to us or dangerous. All these side effects were transient, after a day or 2, they tended to go away. Let's see, that's probably what I wanted to say about that. I think the takeaway from this is that this is definitely well tolerated and quite safe. Also importantly, and I don't mention it here, but one of the risks of ketamine is that you really can increase heart rate and blood pressure. We didn't see any real changes in vital signs, unfortunately. We looked at a lot of different symptoms. We were interested in autism, symptoms of things like social withdrawal. We used a whole variety of measures, both collected from parents, but also based on sort of clinician judgment, using something called a visual analog scale. And we looked at the effect after a week, 2 weeks, and 4 weeks, and we saw benefit really across the board. And interestingly, that benefit sustained itself over the course of 4 weeks. Even a single infusion 4 weeks later continued to be associated with some benefit. Again, all the parents who brought their kids in knew they were getting the medicines. There was no placebo control. That obviously creates an enormous amount of expectancy bias. But nonetheless, we saw a fair amount of change and a lot of stories that seemed pretty convincing in terms of the potential benefits. And it's interesting that we actually saw benefits in kind of core symptoms of autism, including social withdrawal, social avoidance, socialization, and that depends on, these are sort of different names for the same kinds of symptoms, but as reflected in different types of measures. Of course, sensory symptoms are very prominent in ADNP syndrome, and also, included in the autism diagnostic criteria. And we used 2 different measures, the short sensory profile and the sensory assessment for neurodevelopmental disorders that was developed actually here at Mount Sinai. And we saw improvement in both those measures as well. The SAND lower scores are better, sort of more typical, and then the short sensory profile on the left, higher scores are more typical. So that's a little bit confusing, but they're both kind of pointing in the direction that will improve. And then, of course, a lot of these kids have symptoms of attentional problems and hyperactivity. We use a whole variety of measures to try to capture these domains. And we saw improvement, in this area as well. Most of these are parent report measures. That they're all parent report measures. But nonetheless, a lot of kind of convergence. So, it's one thing if you see sort of improvement on one measure, and it's not consistent with other measures that capture the same domain. But here we saw improvement related across the board. I thought that was pretty important. But that was sort of the clinical piece of everything, and trying to kind of think about how these kids are changing in a much more subjective way. We come to accept the idea that parents are experts in their kids, and we as clinicians see ourselves anyway as experts in interpreting parents' experience. But the ideal is to actually have more objective measures, things that you can sort of look at either brain electrical activity or blood-based biomarkers. We've tested a number of different biomarkers, and one of the ones that we use is this thing called auditory steady state response. This is an EEG-based measure. So we know, as I said, that ADNP regulates glutamatergic systems. We know that dysregulated glutamate and GABA, which is the inhibitory neurotransmitter that sits in balance with glutamate. These things are reflected, this balance is reflected in gamma frequency, gamma electrical frequencies in the brain. And the idea that children with autism actually show reduced ASSR, which is consistent with gamma deficits, essentially, has been documented in the literature. And here, what we did, we actually sort of did EEG measures post ketamine infusion, and we saw that there were changes, and that was important to us because we know that these changes are associated with greater sensory symptoms in ADNP syndrome. We know that actually administration of ketamine in animal models and in humans changes this kind of auditory steady state response. This is just a graphical depiction of that, so that the gamma oscillations significantly increased baseline compared to week 1 with ketamine treatment. And that's an important finding. It's a very small sample of kids, but the idea that you can use a objective marker of electrical activity of the brain to potentially predict treatment response is quite important. Thinking about the future, and it doesn't matter necessarily what the medicine is. Ketamine's got an advantage that it likely regulates these gamma oscillations, but the idea that you can take a medicine, and even though there may not be initial clinical improvement, there may be some initial changes that show A, target brain activity, or B, improvement in things like ASSR. Those might be kids that are likely to then go on to respond to ketamine treatment or any treatment for that matter that potentially regulates gamma oscillation. And the figure on the right, this is one of the things that we're working on in the Sewer Center, where we think that the kind of universe of people with autism, even without ADNP syndrome or without a specific genetic cause, they're all very, very different, obviously. And the goal would be to potentially use things like EEG markers to subtype autism more broadly and think, oh, if ketamine helps kids with ADNP syndrome, for example, then it might also help kids with idiopathic autism, where we don't know the genetic cause, but who share different waveforms or frequencies that are picked up on the EEG signals. So these are ways of kind of developing objective measures to predict treatment response. And then finally, Michael Green and Ariel Price, Computer Center, looked at various gene expression studies, pre and post ketamine treatment, and found some additional interesting things. And we were hoping actually to see an increased expression of ADNP, and that would be the ideal. The problem with, I think, the baseline, there was so much variability in the levels of ADNP expression that in the context of this very small study, there wasn't like clear changes in ADNP in particular. But overall, there were very, very clear gene expression studies, gene expression changes, and specifically in immune-related cells, so they're monocyte-related immune cells. Also, it was interesting in this study that they showed that after about a week, the expression changes kind of return to baseline. So the idea that this one-time infusion of ketamine was associated with very clear gene expression changes, but those expression changes were relatively transient. So the figure on the left is showing the differentially expressed genes. The red are, I think, upregulated genes, and the blue is downregulated genes. The figure in the middle is a volcano plot. that shows kind of baseline to post-infusion, and it also shows some specific genes. And then the one on the far right, the heat map basically that shows that most of the genes are actually immune-related. So if you control for immune-related genes, it's actually accounting for about 98% of the signal in the differential expression. So this is pointing to an immune-related function of ketamine, and again, the idea that the effects of gene expression changes are really relatively transient. So this is what you guys are waiting for. All this was very promising for us. I think first and foremost, there's clear evidence that the medicine was safe, at least after a single dose. And there seems to be some pretty good evidence that there's some clinical benefits. A lot of this needs to be worked out, but the idea would be to go forward with a randomized control trial. We'd like to involve at least 30 kids. There's a lot of different ways that one can design this type of trial. But we thought about using a crossover design, meaning everybody would get exposure to medicine and everyone would get exposure to placebo. And you could use both these groups to kind of compare to each other, and that would be advantageous today. The sample, the number of kids who had to participate would be a bit smaller, and it's all powered and objective to drug placebo. And then also everyone would get access to the actual medicine. So the other alternative would be to do, what's called a parallel group study, where you have people that are enrolled in the study that get drug and people that are enrolled to get placebo. But then, not everybody gets drug, obviously. So this is the way we've designed it. There will be a number of clinic visits. One of the big challenges has been how do you actually administer the ketamine? I think you could do it intravenously, you could do it intramuscularly. This study, we chose to do it intramuscular because it's a lot easier and you don't have to do a 2-hour infusion, obviously. We see this as still, very reliable. I think somewhere between 90 to 95% of the drug still gets in the brain based on preclinical study. So the challenge is where do you do it? And so ideally, parents should be trained to actually give these injections at home. But the Food and Drug Administration in the United States did not allow for that. So we need to bring these kids into the actual clinic. This is one of the challenges, I think, in terms of getting funding. And let's see what else I can say about this. So, right, so you'll see the design. Both phases are exactly identical. It's basically 4 weeks in each phase. And then in between the 4-week washout period. So you'll get drug for 4 weeks, washout for 4 weeks, and then placebo for 4 weeks, or the reverse. You'll get placebo for 4 weeks, washout for 4 weeks, and then drug for 4 weeks. And then we'll do a whole series of kind of post-treatment assessments to look at maintenance of drug effects, look at biomarkers, look at gene expression, so on. We would intend to do a whole variety of measures, really try and capture the entire phenotype. So we'll be looking at all the different symptoms that we were just discussing in the open label study. We'll do blood biomarker sampling, and we'll also do some EEG measures. So initially we proposed doing the drug dosing at home. We got put on a clinical hold, as the FDA called it. This is our first light of event. And then eventually we kind of capitulated and we asked and said, okay, well, we'll do the drug dosing in the clinic and the requirements for 2 hours post-dose. Then they agreed to that design. But that's challenging because it means people are going to have to come into the clinic. They get to twice a week over 4 weeks. But now they've approved it. We applied to the NIH for funding and we got a pretty good score on our grant, but it's not going to be funded because it wasn't scored well enough. We have to revise it, resubmit it, and look for other avenues for funding. This is our team. Happy to answer any questions.
Thanks a lot for this update on the trial. And 2 years ago you showed some, but it wasn't this much more extensive analysis of the data now than it was 2 years ago. So thanks a lot. I'm sure there are questions from the audience? Very silent.
All right, so this is the tail end of your 3-day conference, right?
It is. Just a question from my side. You're planning the trial, but as the people have to come to the clinic, I assume it's going to be very restricted to the New York area. Or
Yes, I think, there are ways to hire nursing companies who will go to the home and administer the medicine, remotely. That just, it significantly adds costs.
yeah,
I think, as kind of a pilot study from a funding perspective, this is a kind of a very exploratory study. So the likelihood that someone's going to invest an enormous amount of money in both the trial and the home-based nursing support seems to be relatively low. And yes, you're right. I think that in order to participate and be present in the clinic for multiple doses a week, people would have to like relocate essentially for a month at a time. But it's definitely not, it's not trivial. It's not trivial. I think that's been one of our big stumbling blocks. In that sense, ketamine is not ideal. There's some bias, I believe, about the use of ketamine in general, because people consider it to be potentially a drug of abuse, which to me is unfair and discriminatory. Frankly,
you may want to give it a different name.
Right, or
That will help, just to, like the kind of yeah, no, can support those. No, it's interesting, but it's only, I guess, for the tests that you're doing, they're probably only in English. We can't do them, for instance, in Dutch-speaking countries. So can you?
yeah, so I mean, it's certainly possible to do these assessments in Dutch, but in Dutch-speaking countries, but I think that we would have to just train different sites. There are other sites that are very interested in, and there are sites that are independently, and I've shared this protocol with multiple investigators. We are very happy to collaborate with anybody who wants to get this started. So there's a site in England, I believe, who are looking for their own stream of funding and would potentially replicate this design. So it could be that we kind of create a multi-site study, just using different sources of funding. And then at the end of the day, kind of pull our results potentially. So there's a lot of ways to do this. In rare disorders, we have to be very creative. And we have to be especially creative when we're trying to work with medicine like ketamine.
Okay. Are there any burning questions for Alex, for Dr. Polevson? There's not. I think Alex, we're going to thank you again very much for giving